Hello students, this is Professor C.P.S. Verma, HOD Pharmacognosy and Phytochemistry Department and Dean Student Welfare, Laureate Institute of Pharmacy, Jawalamukhi, Kathod, Himachal Pradesh. Today we will discuss the metabolic pathways and radioisotopes in biogenetic studies, like the overview of the secondary metabolites. So you must understand what is primary metabolites, intermediate metabolites, and secondary metabolites. Primary metabolites are those metabolites which are used for the growth of the plant. Suppose glucose or carbohydrates or polysaccharides. These are needed for the growth of the plant. And where from it will come? It will come from the atmospheric carbon dioxide and water, and in presence of sunlight and chlorophyll present in the plant's leaves. And through the process of photosynthesis, there will be formation of carbohydrates. So, carbohydrates and amino acids and proteins and all these nitrogen containing compounds, these are the primary metabolites which are essentially required for the growth of the plants. So these all come under primary metabolites. So what is intermediate metabolites? Intermediate metabolites, in the enzymology and biosynthetic pathway, there is connection between these two because different enzymes are responsible for splitting or breaking or working as a coenzyme for the formation of different intermediate metabolites. Suppose glucose will split into pentose phosphate. Pentose phosphate pathway is a different pathway and it is involved in the formation of secondary metabolites. But pentose phosphate and erythrose phosphate are used as intermediate metabolites. So next, these metabolites are used for the further pathway. For the cyclic acid pathway, it will further split into phosphoenol pyruvate, and phosphoenol pyruvate will go into the another oxidative decarboxylation, and oxidative decarboxylation of pyruvate will lead to acetyl-CoA. And acetyl-CoA in presence of phosphorylation ATP and ADP, adenosine triphosphate, and this phosphorylation, it will give more energy to go for the further steps. So acetyl-CoA is the basic unit for making or for the formation of mevalonic acid. Mevalonic acid is this is the first step for giving different metabolites, because mevalonic acid it will lead to give secondary metabolites like terpenes, steroid, carotenoids, and anthocyanins, many products. But the research was taken place many years before, maybe 1885. It was searched out that mevalonic acid, it contains 6 carbon atoms, but the scientists need to change it to the 5 carbon atoms. So with the necessary cyclonization, condensation, and enzymatic reaction, they are able to change it to isopentyl pyrophosphate, IPP. IPP is the one which contains 5 carbon molecules, and this IPP is giving rise to isoprene units, isoprenoids. So isoprenoids are the basic units for the formation of terpenes. So after isoprenoid formation, there will be another addition of one more molecule of IPP, it will give geranyl phosphate. Then geranyl phosphate will give C10. C10 molecules. Yani isoprene is the half molecule. So isoprene molecule is having the molecular formula like C5H8. So when two isoprene unit will form, then it will form terpenes, monoterpenes. Monoterpenes means C10H16, which will multiply C5H8 into 2. So it will form monoterpenoids. Monoterpenoids is having C10H16. So diterpenoids to make more C20. So to make the carbon molecules to pile up or to accumulate more carbon atoms like C20, then again there will be addition of IPP, then again there will be formation of geranyl phosphate or fascinyl pyrophosphates. So it will give another 15 C15. C15 will be called as sesquiterpenes. Then C20 diterpenes, C30 triterpenes, like that, and C40 tetraterpenes. So after C40, it will come under like rubber products and gatta and other the solid products will be found. But up to C15, there will be essence. So sesquiterpenes and terpenes are giving good smell, and it comes under with essential oils. Essential oils are having terpenes, monoterpenes, and sesquiterpenes. So like this, mevalonic acid pathway, it will give secondary product like terpenoids, terpenes, steroids, carotenoids, anthocyanins, and iridoids. Iridoids is also one of the category of terpenes. Then aliphatic amino acids, it will give rise to alkaloids. Like that, the whole process of pyruvate and from Krebs cycle, it will come through this and it will enter in the citric acid cycle or the Krebs cycle. Then it will again change into carbon dioxide and water. So like this, the whole process will go on. Another step, if the pentose pathway, pentose phosphate and erythrose 4-phosphate. Erythrose 4-phosphate is a tetrose. You know, hexose. Glucose is having 6 carbon molecules. So it is also called as hexoses. But pentoses, it contains 5 carbon molecules. So it comes under pentoses. But erythrose, it is having 4 carbon molecules and it is called as tetroses. But glyceraldehyde and other things, these are coming under trioses. So further steps, it will change into sigmate. And sigmate will give different secondary products, aromatic compounds like phenyl propanoid, lignans, aromatic alkaloids. These are the different deamination, and if suppose there will be addition of nitrogen molecule, aromatic compounds, aromatic amino acids, there will be different categories, aromatic this alkaloids, then mixed alkaloid, then aliphatic alkaloids. So like this, and acetyl-CoA will also go into another synthetic pathway like polyketides and polyphenols. Polyphenols will be responsible for the formation of tannins, hydrolyzable tannins, and condensed tannic and flavonoids and flavonoids. These are the different steps how the acetyl-CoA will change into the secondary metabolite. Same thing, acetyl-CoA is responsible for the formation of fatty acids. So that will come as acetate malonate pathway. So that is purely for the formation of fatty acids. But mevalonic acetate malonate pathway, it will be responsible for giving rise to terpenes, steroids, and carotenoids. So in acetate mevalonate pathway and acetate malonate. So only purely malonate pathway, acetate malonate pathway is responsible for the formation of fatty acids, like palmitic acid, like adipic acid, like linonic acid, linolenic acid. These are all fatty acids uh, and comes under secondary metabolites. So this is the general overview of secondary metabolites, where the amino acids are also involved. So biosynthesis. So already you studied the biosynthetic pathways is of 4 or 5 types. That is important building block employed in the biosynthesis of secondary metabolites are derived from acetyl-CoA. You know, acetyl-CoA, where from it has come? Try to recall. It comes from glucose, and glucose came from where? From the photosynthesis. It was present in the plant as a primary metabolite, and it will, through the enzymatic action, it will change into pyruvate, and pyruvate will change into acetyl-CoA, and acetyl-CoA, it through the mevalonic acid pathway, it will change into acetyl-CoA. So acetyl-CoA will give rise to different secondary metabolites. Another main important building block is cyclic acid. Cyclic acid also, it will come through the pentose pathway. Pentose hexosid, yani carbon dioxide plus water, it will make glucose or carbohydrates in the plant as the primary metabolite, and then it will further split, it will go into the pentose pathway cycle, and another, there will be formation of cyclic acid, and cyclic acid also we have discussed will give rise to many secondary metabolites. Number 3, mevalonic acid. Mevalonic acid, after acetyl-CoA, next step by step, after the enzymatic degradation, cyclization, condensation, and addition and dehydration, there will be formation of mevalonic acid. And then fourth category is 1 deoxygellulose 5-phosphate. Gellulose 5-phosphate means pentosis. It is a pentose. And then finally amino acids. So primary carbon metabolites, there is chain like this, erythrose 4-phosphate, and again this is the repetition of the same, and the important building blocks are mentioned and shown here, how the mevalonic acid will lead for the terpenes, formation of terpenes and phenolic compounds. And phenolic compounds are nothing but these are all having these tannins, flavonoids, hydrolyzable tannins, like this. So next is acetate pathway. Already we have discussed what are the different steps. We have studied well already. You see, acetyl-CoA is formed by the oxidative decarboxylation of glycolytic pathway product pyruvic acid. Yani when there will be oxidative decarboxylation of pyruvic acid, there will be formation of acetyl-CoA. Yani free oxygen available in the atmosphere is used for this purpose. Not specially it is taken otherwise. So next important secondary metabolites found from the acetate pathway include phenols, prostaglandins, macrolide, antibiotics, and so many. Shikmic acid pathway. Shikmic acid pathway also simply it will come also from the primary metabolites, yani glucose, and it will go into the pentose cycle, and pentose cycle will give rise to shikmic acid, shikmate or shikmic acid, and another side it will go into the pyruvate cycle. Pyruvate will through the oxidative decarboxylation will change into acetyl CoA, and acetyl CoA is the first step to make formation of the mevalonic acid, and different secondary metabolites are formed through this pathway. So you should make it clear what are the different. The shikmate pathway lead to the variety of phenols. All tannins are phenolic compounds. Cinnamic, cinnamic acid derivative, lignans and alkaloids. So these are different. This is the cycle. It is shown in the diagram. Next cycle is amino acid pathway. Amino acid pathway, as you know, these are the primary metabolites used for the plant like peptides, protein, and these amino acids, these alkaloids, and many antibiotics are derived from amino acids. Intermediates from the glycolytic pathway and the Krebs cycle are used in constructing many of them. Because the protein will degrade and peptides will degrade, and it will give rise to amino acids. It can be amino acid can be two types: aliphatic amino acid and aromatic amino acids. So it will give different products or different secondary metabolites like the aromatic amino acids phenylalanine, tyrosine, and tryptophan are themselves products from shikmate pathway. So basically, the shikmic acid pathway is there will be the precursor of phenylalanine, tyrosine, and tryptophan are the basic precursors for the formation of shikmate or shikmic acid. So shikmic acid pathway will lead to all secondary metabolites which are already mentioned, and we can see the steps involved in the diagram. Then we will see how the role of isotopes for tracing the metabolic synthetic metabolic pathway, how we can trace it. Like various intermediates and steps are involved in biosynthetic pathway in plants can be investigated by means of following techniques: use of isolated organ, grafting methods, use of mutant strain, tracer techniques, enzymatic studies. So these are the steps. You see how clearly he is mentioning various intermediate and steps involved in biosynthetic pathway in plants. Whatever the biosynthetic pathways are involved in the plant, that can be investigated, evaluated, they can be interpreted, and by the means of these 4 or 5 methods. Then in our syllabus only tracer technique is involved. So we will discuss this. The label compound can be prepared by use of two types of isotopes. Radioactive isotopes. There will be emission of radiation. If you will decay, then there will be emission of radiation, and that is called radioisotopes. So like a hydrogen and 14C, 24Na, 42K, 35S, like that. For biological investigation, carbon and hydrogen is used. For metabolic studies, sulfur, phosphorus, and alkali and alkaline earth metals are used. For studies on protein alkaloids and amino acids, labeled nitrogen atom give more specific information. 3H compound is commercially available. So this is the isotopes. radioactive isotope of hydrogen 3H. Tracer technique. What is tracer technique? You should know basically what it is. It can be defined as technique which utilized a labeled compound to find out to trace the different intermediates and various steps in biosynthetic pathway in plants at a given rate and time. Suppose we discuss cyclic acid pathway. Cyclic acid pathway, suppose we came to know what is the basic unit to make cyclic acid, that is phenylalanine and tyrosine. Phenylalanine will change into tyrosine and tyrosine will further change and so on by the enzymatic action there will be formation of cyclic acid. So, if we'll use radioisotope of carbon and hydrogen, then we can know, and if we will incorporate these radioisotopes in the biosynthetic pathway, then we can know what are the different steps or intermediate products, intermediate metabolites or products are formed. We can know through the radioisotopes, through the tracer technique of incorporation of radioisotope. In this technique, different isotopes, mainly the radioactive isotope, which are incorporated into presumed precursor of plant metabolites and used as marker in the biogenic experiments. Just now I mentioned you what is presumed precursor. Presumed precursor, as I told you, cyclic acid formation is from the precursor of phenylalanine. Basic nuclei, basic moiety is phenylalanine for the formation of cyclic acid. So if we labeled radioisotope is used, if this labeled isotope is used in phenylalanine, then we can trace the radioisotope, investigate or evaluate what are the steps, intermediate steps are going on. So this is the tracer technique. Then significance of tracer technique. Tracing of biosynthetic pathway, for example, by incorporation of radioactive isotope of 14C into phenylalanine, the biosynthetic cyanogenetic glycoside prunosine can be detected. Understand? So, basically, what the cyanogenetic these glycosides are formed through the phenylalanine through cyclic acid pathway. So, if we will incorporate radioactive isotope of 14C into phenylalanine. So whenever the steps will go on, we can trace easily what are the intermediate products are formed, what are the biogenetic pathway, or what are the different biogenetic this intermediates are formed at different stages and rate and time. Then location and quantity of compound containing tracer 14C labeled glucose is used for the determination of glucose in biological system. Different tracers for different studies, for studies on alkaloids, protein, nitrogen and amino acid, labeled nitrogen gives specific information than carbon. For terpenoids, oxygen atom and glycosides, oxygen, nitrogen, sulfur and carbon atoms are used. Very clear. He is mentioning if we'll go for this alkaloids, this tracing of alkaloids, then protein, nitrogen, proteins and nitrogen amino acids, we will use the label nitrogen. And for other biological activities, we will use the label carbon and hydrogen. But for the different glycosides, we will use oxygen, nitrogen, sulfur and carbon. So convenient and suitable techniques. Application of radioisotopes in biological science. Radioisotopes are frequently used for tracing metabolic pathway. Mixing radio-labeled substrate and samples of the experimental material and collecting samples at various times various times, extracts and separate the products by chromatography. There will be this is the chromatography is the separation technique used. It's the latest advanced technique for separation of different products. Thank you.